Good afternoon. Thanks for being here. So I'm here from Demetrix today to tell you about how we are unlocking access to rare cannabinoids using scalable biosynthesis. So our mission at Demetrix is to make nature's rarest ingredients accessible, sustainable, and affordable for happier, healthier lives. We focus on natural products because they have huge impacts and huge challenges. And so I've got three of my favorite plants on this slide. On the left, you have Artemisia annua. And that's the plant also known as sweet wormwood, from which the anti-malarial drug artemisinin is derived. So artemisinin is the drug that's recommended by the World Health Organization for treatment of malaria in the developing world. So it's a very important drug, and we need a lot of it. And it's also a drug that has suffered from huge price volatility. So swings of as much as tenfold in pricing. And that occurs because of boom-bust cycles in agriculture, because of bad weather, because of stockpiling and speculation. So a few years ago, a second source of the drug came online. It's a semi-synthetic form. So basically what happens is that scientists engineered some yeast with the genes to make a precursor of artemisinin, and then that precursor can be converted by human chemists into artemisinin with a couple of more chemical steps. And so what that's done really is put a ceiling on price swings for the drug, and it's leveled out pricing considerably and made the situation more manageable. So the second example that I wanted to bring up is Taxus brevifolia, or the Pacific yew tree. And that is our source, or was our source, of the drug Taxol, which is an important cancer drug. The Pacific yew is also an endangered species, and so you can imagine that that was not going to cut it for supplying a cancer drug. And so again, a semi-synthetic, actually several semi-synthetic methods of producing Taxol have been developed, and that has led to the ability to supply this drug to the world, essentially. And of course, the last example I'm going to talk about is cannabis. And so, with the approval of Epidiolex recently, that's a botanical extract from the plant, so a highly pure preparation of CBD. And of course, the excitement about, how many other cannabinoids that are even rarer, I would propose that there could be a scaling challenge ahead of us for many of these compounds. And I would also propose that, again, the solution could be production via fermentation, maybe with some semi-synthesis. So today's market for natural ingredients across the board is putting considerable pressure on traditional harvesting methods. Consumers are demanding effective and rare natural products, and these are global consumers now. Brands are requiring a stable supply of high quality and high purity ingredients to meet that demand. On the pharmaceutical side, botanical drugs are extremely difficult to manufacture and scale. There's very few of them, actually, that have been approved by the FDA. And so we think across the board that agriculture is going to struggle to meet the growing demand for these types of very rare natural products. I think everyone here would also agree that cannabinoids offer a range of untapped opportunities. And this is a chart from Acro Capital forecasting legal cannabis sales in the United States through the year 2030, as well as the number of consumers. And so they're making the assumption that by 2027, cannabis will be legal federally in the US. And of course, no one knows that. When or if it will be legal. But I think that, taken together, looking at kind of loosening regulatory structures in the US and abroad in the last few years, there's reason to be optimistic that we could really have 60 million consumers of cannabinoids in the US by the year 2030. And so if that's the case, the question that occurs to me is, which cannabinoids will they be and where will we be getting them from? So our understanding of cannabis is growing beyond THC and CBD. As we've heard in several talks before today, there's more than 100 different cannabinoids that have been at least identified or structurally characterized, and we just know very little about them on the whole. And I would say one of the reasons we know so little about the majority of these is that the majority of cannabinoids are virtually inaccessible in the plant due to their scarcity. So this isn't the case for the common cannabinoids like THC or CBD, which make up anywhere from, let's say, 10 to 30% of the dry weight of the flower. But it really drops off quickly after that. So you start talking about the varins like THCV or CBDV, or CBG, which is the precursor to these other cannabinoids, and those are more like 1% of the dry weight in the flower of the plant. So that's putting you closer to $100 per gram cost to isolate and purify these. And from there, you get into the rare cannabinoids, which are 0.01% to 1% of the flower dry weight. That's $100 to $10,000 per gram, and that group includes CBC, by the way. Finally, you get into these very rare cannabinoids. These are probably structures you haven't seen before. Those are less than 0.01% of the flower dry weight, and that means that they would cost, if anyone was doing it, greater than $10,000 per gram to produce. So it was really this challenge that motivated our founder, Jay Keasling, to establish Demetrix in 2017. So a little background on Jay. He's a busy guy. He's a professor of chemical engineering and bioengineering at UC Berkeley. He's also the associate lab director for biosciences at Lawrence Berkeley National Labs, and he's the CEO of the Joint Bioenergy Institute. He's generally regarded as an authority in metabolic engineering, and he's founded multiple companies. So down there you can see LS9, Lygos, and then Amyris, which is actually the company that developed that artemisinin technology I was talking about earlier, out of an invention that came out of his lab. And it was really a breakthrough discovery that Jay made with his lab that provided Demetrix with a real head start in this microbial production field. At this point, we've raised over $60 million in funding in seed and Series A based on the strength of Jay's reputation, the team that he's been able to put together, and the progress we've made in the last 2 years. Our major investors are Tuatara Capital and Horizons Ventures. So I'm going to start talking a little bit about what the plant does to make cannabinoids, because everything we do is inspired by the plant. And so it's a very interesting pathway in plant. Interesting means hard when it comes to metabolic engineering. So starting over here is a metabolic pathway that kind of shows how the plant pushes chemicals to make the final cannabinoids down here. And so at the top you have hexanoic acid coming in. And through a series of conversion steps and incorporation of other cellular metabolites, you get down to this molecule, olivitolic acid. Olivitolic acid can be joined together with a second molecule called geranyl diphosphate, or GPP, to make CBG A. And that, of course, is kind of the mother cannabinoid, the one which all the others are made off of. And the enzyme that does that is called an aromatic prenyltransferase. And I'm going to talk about that a little more later, so keep that one in mind. Once you have CBGA, that can be converted into delta-9 THCA or CBDA through the action of these cannabinoid synthases, so THCA synthase or CBDA synthase. There are other synthases as well. We heard about some of them today, many probably uncharacterized. And then finally, the A here, of course, means acid, and they can be decarboxylated by heat, for example, into THC or CBD. And so what makes this pathway unusual and more challenging for us is that it occurs in 2 phases in the plant. So in the first phase, GPP and olivitolic acid are made inside the cell and joined together to make that CBGA molecule. That's where it gets interesting. So the CBGA and the synthases are then secreted into an extracellular kind of lumen, a chamber called the trichome. And so that last reaction to make THCA or CBDA is occurring outside of the cell. So I have a weird observation now. It's occurred to us that about 1,000 cannabinoid-producing yeast cells could fit into a single trichome. And so if instead of doing that, we were to fill up a large fermentation vessel with those yeast cells, we'd essentially have a gigantic trichome. And that's kind of the point of all this, is that fermentation in Demetrix has the advantages of a specialist. So if you think about a plant is an amazing organism, and it does a lot of stuff. It grows roots, it grows leaves, it does photosynthesis, it makes thousands of compounds. And in the cannabis plant, in a very small part of the total biomass, it also produces these trichome structures and makes the cannabinoids that we're interested in. So that means that it's actually inefficient for some of these rarer natural products because it's doing so many other things. So it requires a lot of resources. We have limited control, even with some very heroic breeding, over the final products it makes. And in the end, you have a lot of waste, and the process overall isn't very agile. And so by contrast, fermentation is really a specialist. So it takes a streamlined and highly efficient approach, requiring fewer resources, complete control, and creating little waste and working quickly. So I want to get into this in a little more detail. Basically, as a specialist, we take nature's unpredictability out of the equation. So the process that I'm describing today is consistent and pure. It's resource light and reliable. It scales easily and quickly, as the market does. And it yields ingredients in weeks instead of months. So if we were to follow the production of some purified cannabinoids through via agriculture or fermentation, that comparison would look like this. So if you wanted to make a lot of purified cannabinoid via agriculture, you'd start by growing a lot of wet plant, say 100,000 kilograms. And that's going to be probably 3 to 4 months at minimum to get your plants grown. You then dry down the plants. Now you're at 30,000 kilos. You trim away to get just the flower. Now you're down to 10,000 kilos. And then you do some pretty intense purifications to separate these cannabinoids from all the other products in the plant, from the waxes, the other chemicals, the tissue. And in the end, if you had a high THC strain, you might have 2,000 kilos of THC. If you had a strain that specialized in making CBD, maybe you're at 1,200 kilos of CBD. If you're going after one of the rarer ones, like the varins, CBDV, you'd have only 250 kilos. And if you're going after one of these really rare compounds I was talking about earlier, I'll call it CBDX, you'd have as little as 1 kilogram. And this is really a best case scenario. It's going to be variable depending on weather, geography, and of course the natural abundance of the compound you pick. So if you were to take the fermentation route, you would inoculate a large fermentation tank with the cannabinoid producing yeast. They would fill up the tank by growing over the course of 1 to 2 weeks. And then that would be it. The fermentation would be done. And so at that point, you would do a purification process, which would be much more straightforward than purifying from plant tissue, because now you're just separating from yeast cells and from the media in the tank. And you would end up with 2,000 kilograms of either THC or CBD or CBDV or the CBDX rare compound. And that's because the fermentation process is not dependent on the natural abundance of the compound in the plant. We choose the strain that we grow, and it makes the product that it's engineered to make. It's also reliable. This is not happening outdoors in a field. It's independent of weather, geography, and as I mentioned, the abundance of the compound. So I'm going to get into a little more detail here. I'll try not to get too hardcore because it's the afternoon. But the Demetrix process to produce, I'll call them common cannabinoids, THCA and CBDA, from engineered yeast looks something like this. So we start by engineering a yeast cell over here. And so this is Saccharomyces cerevisiae. It's baker's yeast. It's what brings us beer, wine, bread, pretty well-known organism. We then basically need to bring in all the parts to make cannabinoids. And so I like to think of it like an assembly line, where each gene we bring in, each piece of genetic engineering we do is a unit operation in this assembly line. And so we source those unit operations from whatever organism has the best version, basically. And so some of the genes in our yeast come from cannabis, but some of them come from other organisms. So in the end, we have this engineered yeast cell, and we're going to feed it sugar, and we're going to feed it hexanoic acid. And so the yeast cell takes the sugar and converts it into geranyl diphosphate, that's that GPP molecule, and another molecule called malonyl-CoA. In combination with the hexanoic acid we feed in, the cells can make that olivotolic acid molecule I talked about, and then finally make the CBGA molecule. From here, if the strain has a CBDA synthase, we make CBDA. If it has a THCA synthase, instead we're going to make THCA. So these are common cannabinoids, but they're obviously valuable products that do important things. So they go into APIs, which are pharmaceutical precursors. They go into consumer products, anything from skin creams to nutraceuticals, and of course there's the recreational market with THC. So I think where it gets really interesting, though, for this conference is when we replace this hexanoic acid with different carboxylic acids and start to get access to rare and new-to-nature cannabinoids. And so, for example, if we were to take that same setup and feed butyric acid instead of hexanoic acid, now we'd be making varins. So we'd make CBGVA, which is the varin form of CBG, and we'd make CBDVA, which is the varin form of CBD. We can go, so that's basically a carboxylic acid with a shorter chain. We can also go to a longer chain, like octanoic acid. And so in that case, we'd be making these compounds that have C7 tails, both the CBGA and the CBDA version. And by the way, if I had a THCA synthase here, we'd be making the THCA versions of these compounds. The last thing we could do is we could go to the Sigma catalog, and we could select 30 carboxylic acids that look roughly like hexanoic acid, but obviously are pretty diverse there, and then just test them in our strains. And we've done this, and we see that the majority of them end up getting incorporated into the final molecules. Those little blue stubs there represent any of these functionalities. And so now these are not natural compounds anymore. We're calling them new-to-nature cannabinoids. And of course, these are very interesting for new drug development. You can imagine, given the number of carboxylic acids that are available, that you could make quite a few compounds this way. So I want to zoom back and just kind of summarize the advantages of this system. Basically, we have an agile platform for the economical production of rare cannabinoids, rare and novel cannabinoids. And so we have two dials that we can turn on this platform. The first is the strain, the yeast strain. So for example, we could choose a strain that makes CBD here. And so we fill up the fermenter with those yeast cells. And then we choose the feed, the carboxylic acid. So if it was standard, we'd be feeding hexanoic acid. And in that case, we're going to make CBD A, which is the natural form. It's chemically identical and indistinguishable from what the plant makes. However, if we were to switch to a THC strain over here and then instead feed a rare carboxylic acid, so that butyric acid example I was telling you about, now we'd be making the varin form at the same efficiency that we make the natural molecule or the more common molecule. So getting to where we could do this, there were some challenges along the way, and I'm going to go through the kind of big 3 challenges we faced and at a very high level how we've solved them. And so the first challenge was really debottlenecking CBGA production in yeast. And so CBGA, remember, is what you get when you combine that olivitolic acid and GPP molecule with an enzyme called an aromatic prenyltransferase to give you CBGA. So when we first started out using the enzymes described in the literature, we were not making a lot of CBGA. Excuse me. So what we did was put in the enzyme that the Keasling lab had identified called CSPT4 and immediately saw a massive improvement in production. So greater than 10,000-fold improvement in production of CBGA. This is pretty important because no CBGA, no downstream cannabinoids. So I wanted to spend a little more time on that enzyme. And so the original characterized enzyme was called CSPT1. This was discovered by Jonathan Page's group. And it's a relative of CSPT4, but actually fairly different, only about 65% identical. And CSPT1, I would say, cruelly makes, well, for the microbial engineer, I can't speak to the plant, but for the microbial engineer, it's a cruel enzyme because it makes just enough CBGA in yeast that you might think, hey, this is the right enzyme. It's just we've got to adapt it to the yeast cell. We've got to tweak it to get it into the yeast cell. But I guess no amount of tweaking has really worked well there because groups have moved on and found another alternative, NPHB. So that's another aromatic prenyltransferase from Streptomyces bacteria. And that one makes, well, the wild type version of the enzyme, the natural version of the enzyme makes mostly the wrong product. But if you do some clever mutagenesis on it, which has been done, you can make a modest amount of CBGA. Lastly, we get to CSPT4, and that's the enzyme that the Keasling lab developed. And when you put that into yeast, you can just make, I would just say, massive amounts of CBGA. And so it's pretty clear to me, at least from the microbial work, that this is the enzyme that is meant to do that step. And to use an engine analogy here, CSPT1 is maybe a compact car engine, NPHB could be a truck engine, CSPT4 is more like a jet engine. So we found ourselves in early 2018 in the position of having strains that made a lot of CBGA, and that's where we ran into our second problem. And so that was that the cannabinoid synthase enzymes just don't work well in yeast. And so remember, we're making CBGA, we got past that hurdle, and now we want to convert that into CBDA here with one of the cannabinoid synthases. In this case, we're trying to use CBDA synthase. And so it basically totally failed at first, like barely detectable production, micrograms per, nothing. So at that point, we took kind of a 2-pronged approach. So we re-engineered the synthase enzyme itself to try to make it more amenable to yeast. And then we also attacked from the other end and re-engineered the yeast cells to make them support the function of this enzyme better. And so that actually was quite a few steps. So you can see this was the synthase optimization early on, and then there were a series of engineering features that we stacked to get to the point where we were essentially converting all of the CBGA our strains was making. So the third challenge is scale up in manufacturing. So basically, 2019 has been all about getting those strains into fermentation vessels, so basically tanks that mimic what they would be in at full-scale production and manufacturing, making sure that we have processes and strains in place that are stable, and that's in process. And that sounds less interesting and maybe less hard than the first 2 challenges, but for folks that have been in the industrial synbio area, they know that this is actually quite challenging. And the good news is that we have several folks on our team who have experience with that. I think there's over nearly 10 molecules that have been scaled to multi-ton amounts in production. And that experience is in our team. And so, we take this challenge seriously, but we're also confident we can do it. So, in total, I've really told you about how we've debottlenecked microbial cannabinoid production in yeast. And so what that means is we have a platform where we can produce the common cannabinoids, And then we can also produce rare and new-to-nature cannabinoids. So we're using our funding right now to go further faster, and there's kind of 3 parts to that. The first is the team, and at this point we're up to nearly 35 people, and it's a team of experts in R&D, in manufacturing, commercialization, and in regulatory, of course. We've also invested in building a best-in-class lab, and so I'd characterize it as a lab that's efficiently focused on production of strains to make high-value natural products. And so there are a lot of automation and computing tools that are a part of that lab that make the work go faster. And so essentially our scientists spend a lot less time pipetting, and they can kind of do higher value activities. The last piece that I think is pretty important too is that we're developing industry-leading intellectual property around this microbial production. And so we've invested in IP licenses, for example, the license of the work from the Keasling lab. And then over the last couple of years, we've also developed our own suite of intellectual property that is propelling our technology forward and enabling commercialization. So with that, I wanted to return to our mission statement, which is to make nature's rarest ingredients accessible, sustainable, and affordable for happier, healthier lives. And really everything that I've been talking about in the last 20 minutes has been about the first part, the accessibility part, the ability to scale these compounds. And what's great about being at this conference is getting a chance to see the second part, which is finding out what these compounds are good for and how they can really impact human health in meaningful ways. And so with that, thanks for your attention, and happy to take some questions.
Hello there. That is absolutely fascinating work. I love seeing the adjustments on the prenal transferases. I'm trying to get a gauge of the relative efficiencies next to cannabis, and obviously you're using sugar, and 99% of the sugar in the world is made by the Calvin cycle and photosynthesis, and so why rip out the middleman there, and are you getting close to the efficiencies and the carbon audits that make this type of activity economically feasible?
Yeah, I think that yeah, I don't want to knock the plant. I hope it didn't come off like that. I think it's mostly that the cannabis plant has a number of responsibilities. So I can't get into specific numbers on where we're at right now, but I would say the cannabis plant is doing all sorts of things. If its sole job was to convert the sunlight into cannabinoids, it would kick our ass. But it is doing all sorts of things as a plant needs to. And so I think that really what we do is we take a single slice of all the metabolism that plant is doing, put it into yeast, feed them sugar, just run them at full speed, and get the process done in 1 to 2 weeks. And so I think that's where the competitiveness comes in.
Could you comment on the regulatory structure, which licenses you need, and then which labeling would go on the products using those ingredients?
I think so. Regulatory is something we understand is definitely very important and a major challenge in this area, and I am not qualified to tell you the answer to that yet, but it's definitely something we've made central to our early development effort. We're not waiting till we have the product to do it. Is
it a DEA license because it's not a plant?
Yeah, so we're DEA licensed. Everything we do is within federal law, and it'll always be that way. But I think, that situation is kind of changing rapidly, and it's ill-defined right now. So we err on the side of caution there.
Thanks.
Sure.
Thanks for the great talk. You mentioned that you're in the scale-up phase with scaling up the amount of product that you're making. I've seen in literature in the past that there's issues with cytotoxicity with some of the cannabinoids that are produced, at least the terminal cannabinoids. Is that something you've addressed yet in scalability or have seen one way or the other?
Yeah, so it's interesting in metabolic engineering. I've heard this for a number of products, and there are some cases where the products are truly toxic. And in other cases, it just has to do with misbalancing the metabolic pathway. And so I can't get into detail, but I'd say we think it's the latter. We're not overly concerned about the toxicity of these compounds to yeast.
Excellent, thank you.
Yeah. All right, great, thank you.
I think we have one more question.
Oh, I'm sorry. Flee too early.
Check.
What's the yield of the product you are making?
Yeah, I can't tell you that right now. I'm sorry, but yeah.
Yeah, just curious, when can you give us any sense of when this technology might come online and be a competitive threat to the traditional art of growing marijuana and cannabinoids?
Yes, I think first, this is going to be a massive ecosystem, and I don't think that what I described here today is ever going to be a threat to large number of people that want to interact directly with the plant. That's a different experience. I think that we could be a threat to rare cannabinoids pretty quickly. But I don't view it as a threat. I view it more as a bonus because there just isn't a good way to scale those right now. But yeah, I don't know exactly. I'm more on the science side. I don't know exactly how much I'm allowed to say about our scale-up timeline, but it's what we're working on right now, essentially. So
just one other question. In terms of relatively quickly for rare cannabinoids, are we talking 12 months? Are we talking 5 years, 7 years type of thing? Is there any type of time frame?
Definitely less than 5 years, yeah.
Okay.
Do you know, or is it known, what it is about cannabinoids that are toxic to yeast?
Yeah, so I actually don't think they're that toxic to yeast, and so I have seen those reports. I think that that's a hard question to answer. I mean, I guess you could add them exogenously to a yeast culture and maybe see toxicity, but they're not very soluble compounds, so there's questions there. I think that producing cannabinoids can be toxic, perhaps, even if the final. I just fall back on kind of a variation on my last answer, which is that you need to get everything lined up right. Going back to that assembly line analogy, I think of the I Love Lucy part where she's on the conveyor belt with the chocolates and they're basically coming in too fast and starting to spill over. So in metabolic engineering, when you end up in that situation, that spillover instead of being chocolate is usually toxic. So it's that kind of thing where you just basically need to get things balanced correctly.
So if somebody had 3 cannabinoids and they wanted to go into the FDA with a plant and we wanted to start a master drug file, how soon could you do that?
I think it depends on which cannabinoids they were. I think that our technology is particularly amenable to certain kinds of variants. I think that we should have access to, in principle, the full range of cannabinoids from the plant. But I'd say I basically would need to see the compounds to give an estimate. For some, it could be very quick. All right, thank you.